Okay, good afternoon. I would like to thank the organizers for giving me the chance to be here today, and also apologize if I don't sound very focused. It's been bittersweet. It's great to have such a amount of great scientists around. It's not that great that Pepe is not here to enjoy you. My talk is entitled Bacterial Genetics, a classical tool to reveal avant-garde strategies against resistance and virulence. But it very well could have been genetics, the power of classical genetics. I had a slide showing you how important is antibiotic resistance, but Bruno did a great job showing you that already, so I'm going to skip this one. And if we go to the basic oversimplified level, which is the one that I understand, and from an evolutionary perspective, there are two factors that are critical for the generation and dissemination of antibiotic resistance. And these two factors are the rate at which it doesn't work, okay. The rate at which the antibiotic resistance are acquired, and also the fitness cost that they cause on the bacteria. So this is obvious. Then there's a number of accessory factors that the environment has a role in both of these factors. There's a epistasis that is basically interaction between the resistances and the genetic background where they occur. There's also compensation, that is basically the acquisition of additional mutations that compensate, that alleviate the fitness cost caused by resistance. And then there's multiple type of interaction between these factors. So today I'm going to focus on the cost a bit and how we can use the cost in our advantage. I will talk a little bit about epistasis because that was what led me to the lab of Isabel Gordo in Portugal some years ago. They were studying two antibiotic-resistant mutations that are targeting the RNA polymerase and the ribosome. So rifampicin targets the RNA polymerase, and mutations in one of the subunits of the RNA polymerase confer resistance to rifampicin, and streptomycin targets the ribosome, and mutations in one of the subunits of the ribosome cause streptomycin resistance. And before I arrived to the lab, they had discovered that these mutations are highly epistatic, that whenever these two mutations are separated in the genome, they have an effect. But when you put these certain alleles of these mutations together, the effects are not what one would expect. There's higher cost than expected, there's lower cost than expected. There's some epistatic interactions there. So when I joined the lab, I was working in a different project that she had funded and so on. But then one night at 9 a.m. with some beers and so on, I told her, look, Isabel, what I really want to do is this. And then she said, okay, I don't think you're going to get anything, but you are welcome to try. So she allowed me to give a try to this, and that's what I did. So basically, these two machineries, transcription and translation, the RNA polymerase and the ribosome, are involved in two essential processes of the cell, transcription and translation. And the result of that is protein synthesis. So the obvious explanation to interactions and to the cost of resistance mediated by this mutation was protein synthesis. It makes sense. You have mutations that alter the process of protein synthesis, and that causes a cost. And that was explained at the general level, at a global scale, across the genome, or in specific regulons that are very critical. So that was the classical explanation, which makes complete sense. It was clear that that was part of the explanation. Then we had some indications that this was not only the only mechanism behind this cost. One of them was this weird epistasis. It's not very easy to understand that if you have a mutation that reduces the processivity of the RNA polymerase by 10%, and then a mutation that reduces the processivity of the ribosome by 10%, you put them together and suddenly you reduce the fitness cost by 40%. It's not easy to understand that if at the end of the day they are all working on the same mechanism. So that we thought that there was additional factors there. So our hypothesis was that there are unknown factors that are contributing to the cause of rifampicin and streptomycin mutations. And then, since I was coming from where I was coming, I resorted to genetics. And I'm doing my own definition of genetics. This is not a quote. It's the result of many conversations with Pepe, and probably not only with Pepe, about why we like genetics and why genetics is useful. And the summary of that is that the beauty of bacterial genetics is that you can ask nature any question in a way that probably is going to give you an answer, and the answer is going to take you anywhere. You don't know where are you going to land when you ask questions using genetics. With every gene appears, suddenly you are studying something that you were not planning, and so on. So at the end of the day, the answer was not that unexpected in this case. What we did, it was a selection. Now it's called experimental evolution, but it's basically a long-term selection. You get in this case, we got a genotype that has a very large fitness cost. This is like over 40%. It's a double mutant that has a rifampicin mutation and streptomycin mutation. So, as I told you before, if you allow this guy to progress, to propagate in the absence of antibiotic, some other mutations are going to appear that are going to compensate for that cost. So that's what we do in genetics. Let's allow this guy to propagate, and let's find out how it gets to solve the problems that they have, and then we can figure out which were the problems in the first place. And that's what we did. So we did basically 22 days experiment in which we were propagating every day in rich medium in the absence of antibiotics. And at the end of the experiment, we had several lines in parallel, and we did a population sequencing, which reduces the definition, but allows to increase the scope. You can get many mutations that appear in different populations, so you probably miss some of the mutations that are at a low frequency, but you get a bigger picture of what's going on. And then we found several mutations. We found compensatory mutations that were described to be compensating for the problem of the rifampicin resistance. We found mutations that were known to compensate for the problem of the streptomycin resistance. We found mutations that were not described as anything. Some others were just adaptation to the growth medium that happens always in this experiment. But then we found a category of mutations that seem to be specific to this double mutant. When we move these mutations to either the sensitive background or either of the single resistant mutants, were not beneficial, were only beneficial in this background. And these mutations were in particular machinery that is involved in the interaction between transcription and translation. So there are two proteins. One of them you already have heard from Nello, NASG, that is a terminator, anti-terminator elongation factor that connects physically the RNA polymerase with the first ribosome that is coming to translate. And we found mutations that were increasing the expression of this gene. So it was a mutation in the promoter that was causing an increase in the expression. And then we found also mutations in this protein that is called NAS-E, that is a ribosomal protein that interacts with NUS-G in this connection. And we also found this was a mutation in a hairpin that is negatively regulates the expression of this operon. So that mutation was causing also an increase in the expression of this gene. So, and then we found a third mutation here in the RNA polymerase in the region that interacts with NAS-G. So everything made perfect sense. We have now mutations that seem to be involving coordination in between transcription and translation. So our hypothesis was that the problem of rifampicin and streptomycin, sorry, is not only that they cannot produce enough protein, is that transcription and translation are not coordinated. Okay? For the sake of time, I cannot tell the entire story, but we went step by step, finding out that the uncoupling in between transcription and translation is causing increase in R loops. R loops are DNA RNA hybrids. We will talk about it a little bit later. These R loops cause replication transcription conflicts. So the machinery of replication and the machinery of transcription are working all the time in bacteria, and one is faster than the other, so they find each other very often. And if there are R loops, there are conflicts, and these end up generating DNA breaks. We managed to demonstrate all these steps, and then we come to the next point. Can we use any of this knowledge in our advantage? So basically, can we exploit these effects that we have discovered? And then what I did is I have to talk a bit about R loops. That's why I said before. So R loops are these RNA DNA habits that are usually formed in normal conditions during transcription. Is completely normal. They also have very important functions in the cell. For instance, they are important in transcription initiation and termination, in transcription coupled DNA repair, non-canonical replication, plasmid replication is very important. But when they are in excess, it's a big problem for the cell because of the transcription stalling, transcription replication conflicts, and all that I was talking about. So we have a system, these R loops that is involving many things. It's involving evolution because it's controlling repair and so on. It's involving transcription, so it's involving the programs that the cell are activating or not. So it's interesting. It's interesting. And there's a single protein that is called RNase H1 that is involved in removing this RNA from these hybrids. So we have a very specific target that is involved in this business and not many other businesses. So that was interesting as a potential target. So I mutated the RNase H. This is a collection of 16 different susceptible bacteria, streptomycin resistant, rifampicin resistant, and double mutants. So 3 streptomycin resistant, 3 rifampicin resistant, and all the 9 combinations in between these alleles. And what we found this is fitness here on this side. This is a selection coefficient. In black, these are the points, the fitness data in the presence of RNase H in a wild-type background that has the RNase H. In red, you have in the absence of RNase H. If we remove the RNase H in each of these 60 mutants, what you see is that systematically every one goes down in the fitness. And here in blue is the same data, the data of the wild-type data points, but when the bacteria were grown in the presence of this compound that is called RH1001. That is an RNase H inhibitor. Okay, so we have a target that seems to be specific and that causes a very in the fitness of resistant bacteria particularly. Sorry. Probably I should. Okay. ¿Lo apago? ¿Cómo se apaga esto? Vale, ya está. Bueno, so we wanted to know if this had any use. So the first thing that you have to do is to see, will bacteria that are lacking the RNase H really be outcompeted by sensitive bacteria? So we did a very simple experiment. We competed a wild-type bacteria with 5 different resistant mutants, in this case single resistant mutants, because if we use double resistant mutants, they go down in the population too fast because their fitness cost is too large. So we use single mutants, and it's a mixture 9:1, so there's only one mutant, only one wild type per 9 mutants. This is the frequency of mutants at the beginning of the experiment, and we were the same, propagating every day, and then measuring the frequency. Each of them were labeled with CFP or YFP. We could go to a flow cytometer and measure the proportions, and you see that at the beginning, the expectable happens. So sensitive bacteria starting at a very low frequency, but then is taking over, and resistant bacteria cannot do anything to prevent that. But at some point, things start to happen, probably compensatory mutations happen and so on, and then resistant bacteria and susceptible bacteria coexist. And this happens either under a very strong bottleneck when we take very few bacteria to the next propagation, so the chances of rare mutations to pass to the next culture are very low, and also under a mild bottleneck. So those are the red and the blue lines respectively. And then we did the same experiment in bacteria that were lacking the RNase gene, both the sensitive bacteria and the resistant bacteria. And what happens is that in that case is that sensitive bacteria takes over very fast, and these guys never come back. You go to this culture, you play it on rifampicin and streptomycin, and nothing grows. They are dead. They are extinguished. But this is in vitro, and one of the things that Pepe used to say is that in vitro everything depends on the concentration of magnesium. I mean, this is not that much in vitro. There's live bacteria there, but let's go to see what happens in mice. So I was using E. coli in these times, so it's not a proper infection model. K12 garden variety E. coli. It was not a HEPEC or whatever. So we did the same experiment. In this case, we took a mutant that it was known in the lab that colonizes the mice effectively, and we did the same. We did a competition 1-to-1 in this case, and we saw that in the presence of RNase mutants and resistant and susceptible bacteria coexist. In the absence, resistant bacteria fall dead. So then we have an interesting target, but also the resistant threat has two sides. We need to kill bacteria because bacteria are virulent. If we could manage to eliminate that from the equation, if we could convince bacteria not to be virulent, there would not be a problem. We don't even need antibiotics. So there has been some strategies proposed for doing this. One strategy about anti-resistance is what I just showed you. You can try to target certain genes that make resistant bacteria less fit in a population, but then there's a non-biocidal inhibition of virulence trait, the anti-resistance strategy, that is also very interesting. But I told you, RNase is involved in gene expression, so it might be involved also in virulence. Let's check that. So I was at that point when Paco was kind enough to support my application to come back to Seville and join his lab. They are experts in Salmonella virulence. I could go back to work with Salmonella, which was my first love, and I did. So this is a collection of genes. These are luminescent constructions, two promoters of different genes that are involved in virulence. And the conditions are under LB growth, like in the bacterium, or under conditions of induction of the Salmonella pathogenicity island 1 or Salmonella pathogenicity island 2. I'm not going to be describing anything in detail, but you can see that there's quite a lot of changes. This is the fold change in between the wild type and the RNase mutant. So the RNase mutant is actually altering in both ways. It seems to be making crazy the expression of these genes. And as has been said before, it's necessary that genes are expressed in the moment and in the place where they need to be expressed for virulence. Jay was talking about that before. So this could be affecting virulence. I went and tested that. So we don't do mice work, but we do macrophages competitions. I did the typical competition that they talk about, and I found that RNase is actually as attenuated as a double mutant that doesn't have the SPI1 or the SPI2. So I hope, as a summary, that I have convinced you that bacterial genetics is very powerful to identify novel strategies, also that the fitness cost of antibiotic resistance can be exploited, and also that RNase is a promising target for both anti-resistance and anti-virulence. Also, I would like to thank, this is my final message. So we are facing a superbugs now. Bruno explained it perfectly. But I think we can find the kryptonite of these superbugs. And in my opinion, bacterial genetics and evolution is going to make a great deal helping us to find this kryptonite. So it's great that we continue doing it and enjoying it. Also, I would like to thank the lab where I did the work in Portugal and some collaborators there. of course, Paco, who hosts me to come back to Seville. I will be starting my lab in the next few months, dealing about all these processes. And of course, you know that there's one more person that I would like to thank. It's not the boss. Although the boss says something that Pepe and I had in common. So we both love music. Of course, I had a much less refined taste of music than Pepe. And the same for wine and for food and so on. But I love music. I play the guitar. And so Nello, who you know loves to embarrass me, you have witnessed it, used to put a picture of me playing the guitar in the presentations and so on when he was presenting me. But anyway, the boss said music is forever. I really believe music is forever, but I also think that the teachings of Pepe, the generosity of Pepe, the capacity of transmit passion for science of Pepe are also forever. So thank you very much. Thank you, Pepe. And thank you all.